Good morning, students. Today we are going to study about the post-fertilization events in the sexually producing flowering plants. So that is in your angiosperms, we are going to study after the pollination, what is the events. So you have already understood about double fertilization. Till double fertilization is after the pollination events. Just to recall, this is what is your after pollination, double fertilization, and in your post fertilization we are talking about after the fertilization process what happens. You have already studied before, there is the formation of zygote, there is a formation of embryo. Okay, just to recall, these are your previous topics. Zoophily we have completed, outbreeding devices to overcome the self pollination and to enhance the self pollination problems were there, that is your inbreeding depression. To overcome that problem, they have enhanced naturally creating outbreeding devices so that cross pollination is always checked over there. And pollen pistil interaction is clear based on the right type and the wrong type of pollen that can germinate on the stigma of the flower. Then artificial hybridization, it is a man-made. So it is your desirable characters you can select, cross pollinate, and that's how you get your hybrids. So it's all clear with your two techniques in this called your emasculation and bagging. So now under post fertilization events, you have come across previously with two, that is your zygote and embryo formation or embryogenesis. Please note, in plants you have to understand a little deeper. There is first endosperm development and embryo development. Okay, first is endosperm. Please don't get confused with endospore. It is endosperm. Okay, then it is the embryo. Then you have to understand about the maturation step, that is ovule to seed, ovary to fruit. Okay, we'll understand one by one. Before we go into details, let us know what is endosperm and what is embryo in a coconut in general, as a general knowledge. So you all know about the coconut inflorescence. Observe the female part. Okay, this is the female flower. These are all your male flowers. All these are your male flowers. Male flowers fall off. Female flowers grow into the fruit. You can see here. Only stick like remain. That means only male flowers are fallen off. Females have grown into the fruit. This is the one which I have told even last year. When you cut open a coconut, you can find two things. One, a yellow sweet part, and then the white color, okay, which we use for the curry and all, or many things. Okay, so both are edible portions. In this, the yellow portion I had told is the embryo, and the white region is the endosperm. So endosperm is the one which is nourishing the embryo. I hope it is clear. So that is one idea you have got. Now we are going in detail. The endosperm development can be asked for 3-mark question. So you should always remember, this is the process which precedes embryo development. Why does it precede? Meaning to say it is before the embryo development. It has to form before the embryo is formed. Why? Because it has to nourish. As the embryo is forming, to nourish it, endosperm should be present ready. That's why we say it is preceding embryo development. So how does it form? Primary endosperm nucleus undergoes repeated cell divisions to form triploid endosperm tissue. This is your triploid condition. From that nucleus, okay, so there is your cell also formed. Remember, primary endosperm cell. First is the nucleus division inside that cell. Then it is your cellular formation to finally form endosperm tissue. You'll understand that part in the next slide. You should know the significance of endosperm. Since it consists of reserve food material, it is used for the nourishment of the developing embryo. Okay, it has to nourish the developing embryo. So this is the definition. If for 1 mark, if they ask what is endosperm, you should write it is a nourishing tissue that consists of reserve food material for the developing embryo. Okay. So what I said before, now just to recall back, here I've told primary endosperm nucleus undergoes repeated cell divisions. PEN or PEC you can write. First step is free nuclear endosperm. So your nucleus PEN undergoes the nuclear division. So it undergoes successive division to give rise to free nuclei. When I say give rise to free nuclei, it is many in number. So imagine this is your embryo sac. You remember your 3 antipodal cells. Here there is your, wait, let me just give you a clear one. So there is your 3 antipodal cells, egg synergids, and the 2 polar nuclei. So here this is already fused with the male gamete to form the, imagine that is how your secondary nucleus fuses with your male gamete to produce primary endosperm nucleus. Okay, and there is your central cell. Now this central large cell undergoes repeated division. So first is nuclear division. Within this cell itself, the nucleus splits to form many free nuclei. Okay, that is why it is giving rise to free nuclei, many in number I mean. So this is once it has formed. Then cellular endosperm. So first step was nuclear endosperm, free nuclear endosperm. Why free? There is no cell membrane around it. Okay, there is no cell wall around it. Cellular endosperm is, it forms the cell-like structure. So for around each nuclei, there is your cell wall that is producing. So it will start from the edge itself. Okay, so to get a clear clarity, I'll show you a picture. So you can see this is one nuclei, that is your primary endosperm nuclei inside the embryo sac, inside the fertilized embryo sac. Okay, it divides, again freely divides, free division. So this is your free nuclear division. Then you can see the cellular endosperm, you can see the layer of cells. Okay, and for each nuclei there is your cell wall formation. That is why it is cellular endosperm. Similarly, in your tender coconut, if you see, it is watery in nature. Tender coconut is watery in nature. That is what is your free nuclei. Later on, when as it is like the time passes, it becomes mature. As the embryo also grows, you can notice the kernel is also getting thicker. Okay, we can't keep the tender coconut for many days. Definitely it will turn, the kernel will be produced. That is why the white part, the kernel, we say it is your cellular endosperm. Whereas tender coconut, the kernel seems to be very thin, filmy like, okay, soft, which you like to eat. Okay, in that is in your tender coconut. So this indicates there are many number of free nuclei before cellularization. So I hope you are getting the idea. Free nuclear division, that is your number of free nuclei are formed before cell wall is formed. Okay, cellular like structure that is formed. So that is your example, tender coconut water. And you can find your cellular endosperm at the stage of the coconut when it is ready with your white kernel. Okay, So it's let me just show you the picture. This is the white kernel. Okay, when it is tender, you won't find this so thick. You will find it as a thin film. Okay. Now, you should know about something about endosperm. When it forms a seed, the endosperm may be completely consumed by the developing embryo, either completely consumed before the seed germination. Okay, so you can find the examples here. There is your pea, groundnut, and beans. So they are completely consumed. Then the endosperm is completely consumed by the developing embryo. Or it may persist, meaning to say it is remaining. It's like a residual part remaining in the mature seed. So your castor, coconut. So these are all your persistent endosperm. So you might have, you remember you do your kobbari, we say. Only some of you all know. To prepare oil from that coconut, we dry that coconut kernel. So that is your kobbari, right? So that is your persistent endosperm. So it's clear. Please remember, this is not there for your exam. This diagram is not there for the exam. That's why it is only asked for 3-mark question. Not more than that. Okay. Now we are understanding the second step, okay, of post-fertilization event, that is embryo development. Okay. The term embryogeny or embryogenesis, also called as your embryo development. Okay. So this pattern of embryogenesis is similar to both monocots and dicots. So we just observe this embryo sac, which is totally lost its shape because it is after fertilization it starts to differentiate now. So you can find your zygote here, rest all are degenerating here, and you can find your endosperm over here. So see the steps. The zygote is enlarged here. First step, the one division occurs. It is all mitotic divisions here till here. So mitotic division to form proembryo. Zygote to produce embryo. This is the embryo. So first step is proembryo. Then you can see globular embryo. You can see this globe-like globular structure on a stalk. Then there is your heart-shaped embryo. And finally your mature embryo appears like this. Okay. So this is the pattern in case of monocots and dicots. About the structure you will come to know clearly. I've written it here for you for easy. How zygote produces into develops into embryo. The steps are here: proembryo, globular embryo, heart embryo, and finally it is your mature embryo. Okay. So this is your 2-mark question. Please practice it well. So you can find one structure here called as plumule. This you have come across even before the terms. There is a structure here as well as these. If you just see what they have written, these two are cotyledons. This particular portion is a shoot tip, that's why it is plumule. Here it is a root tip, that's why radicle, protected by a root cap. Okay, so let me just tell you one more term. There is, I'll just keep this all straight. Now imagine there is that bump here, plumule, and this is your cotyledons, and this is your hypocotyle, the region below this portion. Now this portion I've marked it here. Let me just mark it in different color for you. So this portion I've marked it here. So there is again a difference. This is your cotyledon, this is your radicle, this is your plumule. Okay. Now I'm drawing an imaginary line. I'm drawing an imaginary line here. It's not straight, I'm sorry. Now imagine this line to be an axis. So what kind of axis is this? Since we are talking about the embryo, it is embryonal axis. I'm just writing embryonal axis since I do not have place here. So just imagine this pink imaginary line as embryonal axis. Here it is curved, that's it. Here it is curved. Now this particular axis is showing plumule is also there, radicle is also there. So that is an axis which includes plumule and radicle. Now always remember, this is the level of attachment. The blue color I had drawn is the level of attachment of cotyledons. Now above the level of attachment of cotyledons, there is a portion. The tip is plumule. The rest of the portion is called as your epicotyle. You'll understand what is that epicotyle. Okay. Don't worry, the spelling I'll write it there. It's difficult to write here. Okay, then the portion below the level of attachment or below the level of attachment of cotyledons, this portion, the cylindrical portion, is called as hypocotyle. Just to be very clear, see here. The portion of embryonal axis above the level of, this is wrong. This is cotyledon. Okay, above the level of cotyledon is epicotyle. Okay, then the cylindrical portion below the level of cotyledons is called hypocotyle. So I hope it is clear. Please note this is cotyledon. Don't write the same thing. Okay. So if the question is asked, describe the structure of typical dicot embryo. Okay, typical dicot embryo. They have told describe. You can write all these points with the diagram. Okay. So these are your points. It consists of embryonal axis and two cotyledons. You need not explain more. You can write the same points. Then you're talking about epicotyle. You're talking about the epicotyle terminates in the stem tip called plumule. Okay, then the portion below is called as hypocotyle, below the level of cotyledons. Okay, hypocotyle terminates in root tip, and the root tip is also called as radicle. So root tip is also called as radicle. Radicle is covered by with the help of a root cap. So this is what we are saying. Epicotyle that terminates with plumule, hypocotyle that terminates with radicle. So it's clear for you. Then coming to your, please draw this many times because frequently asked question this is. So see that you are practicing it well. Okay. Yes. So here observe carefully. Here in monocots only one cotyledon is present. Okay. So this is one cotyledon. The term is scutellum. There are even other new parts here. You can find around the shoot apex. This is the epicotyle part. Shoot apex is here. And there is a covering. That protective covering is called as coleoptile. Similarly here, there is your radicle. There is your root cap. Then there is a protective covering called coleorhiza. So see that you are drawing accordingly. Practice it well. Coming to the description of the structure of LS of monocot embryo. It consists of only one cotyledon. Okay. So what is that one cotyledon called? Scutellum. So if the question is asked, what is scutellum? It is a single cotyledon present in the monocot embryo. Okay, single cotyledon present in monocot embryo. So where is it present? It is situated on one side, that is the lateral side of the embryonal axis. So you can observe here clearly it is only at one side. We are talking about the axis that should be like this. But since there is only one side here, one cotyledon, we are talking about where that one cotyledon is located. It is in the, so since this is the plumule, so imagine this is your plumule, this is your radicle. So embryonal axis will be like this. So it is at this side, one side, lateral side, the scutellum is present or the cotyledon is present. So you all know very well about this part. Lower end of the embryonal axis consists of radicle and root cap. Okay. And it is enclosed, very important. 1 mark question, what is coleorhiza? Radicle and root cap is enclosed in an undifferentiated sheath which is called as coleorhiza. This undifferentiated sheath is called as your coleorhiza. Then this is your all this lower portion below the attachment of cotyledon is your hypocotyle, and the portion above the level of attachment of scutellum or cotyledon is epicotyle. Okay. Then coming to your another, epicotyle has a shoot apex with leaf primordia which is enclosed in a hollow foliar structure which is called coleoptile. So this is coleoptile enclosing the shoot apex with the leaf primordia. It encloses that in a hollow foliar structure. That is your coleoptile. So I'm just going for the next now. Coming to the seed, it is your final product of sexual reproduction. It consists of seed coat, cotyledons, and embryonic axis. So let me just show you first about the seed. You all know about your chickpea, right? Let me just try to draw here. So you might have seen a chickpea like this, not Bengal gram, you all know. Bengal gram. So when it opens, so this is somewhat like your moong gram. Moong, I hope you all know what is moong, green gram. Okay, so there is one portion here and a small portion like this, a dot. Here there is a bigger dot. Okay, so based on that we have an opening called micropyle. This is your micropyle for the entry of water, oxygen for the seed to germinate. Okay, whereas this portion was your old mark of ovule attached to the placenta, that is your hilum. So you don't worry much, that much detail you all are not studying. This is your dicot seed. This is your monocot seed in LS, the longitudinal section. So just remember it has a seed coat, cotyledons and embryonic axis. Okay. So you might have seen when moong gram, the gram, when you soak in water, one green layer comes out in green grams. A green layer comes out, leaving the two cotyledons like this, white color. And then there is a root. So that is your radicle that has turned into root, and then there is shoot formation, that is your plumule that is developing into shoot. White color part is cotyledon. Whatever the green cover that is fallen out, it is coming out, that is your seed coat. So I hope you have understood about the seed coat, cotyledons and embryonic axis. Then cotyledons are thick, swollen structures which reserve food material. Yes. Now it is the cotyledons which are nourishing the developing plant. So you might have seen the cotyledons with the young root and young shoot. Okay. Now there are two types of seed, two types of mature seeds. So this can be asked as a 2-mark question, a separate 1-mark question. Non-albuminous seeds, that is, there is no residual endosperm. Endosperm is completely used or consumed during the development. So that is your pea, groundnut comes in the under this category. Okay, and albuminous seed. So one is non-albuminous, that means endosperm is completely absent or completely used. That's why it is empty over there. In case of albuminous seed, the endosperm is retained inside the mature seed. So which are the examples? Wheat, maize, barley are your examples. Okay. There are even other terms which you should remember. You all know about nucellus. So imagine this is your green gram. Okay, the green gram. So on over that green coat, imagine, I just said imagine, there is certain layers of nucellus. Okay. Now that if it is persistent, it is not able to be removed during the maturation process, then that is your persistent nucellus, which is called as perisperm. Okay. So you can take that as your, yes, the remnants, one and the answer for 1 mark question. Take this sentence only as your answer. Perisperm, it is the remnants of nucellus which are persistent on the seed. Okay, that way you have to write the answer for 1 mark. Then if the question is asked, what is the role of micropyle on a seed? So micropyle in the ovule, micropyle on the seed. You have to know the difference. Micropyle in the ovule, it is for entry of pollen grain. It is an opening. Whereas on seed, it is a small pore which facilitates the entry of oxygen and water in seed during germination. So you all should know the mature seed, okay, the water will be completely reduced or relatively reduced to 10 to 15 moisture content by mass that will be present in the seed. Mature seed. So it is completely reduced. Okay. What is dormancy? It is a state of inactivity, meaning to say all the metabolic activity of the seed or embryo slows down. So when it is favorable, dormancy is inactive, dormant. So when it is favorable, it will germinate. So during that germination process, oxygen, water enter into the seed through the micropyle. Okay, I hope it is clear. What is the moisture content in a seed, in a mature seed? It is 10 to 15% of moisture by mass. Okay, that is a relation by mass they have told. So I hope it is clear over here about the monocot and the dicot seed. Seed viability. So you have already come across with the term viability. We are talking about the life or the validity for the seed, or you have studied before with the pollen. Now let us know that few will lose the viability within few months. Okay, few of them lose it. Some can live for several years. Several years, there is one example here. The oldest lupine, oldest seed, that is lupine, scientific name Lupinus arcticus, excavated, that lupine was excavated in Arctic tundra. A record of 10,000 years of dormancy was recorded. Okay, so this is the oldest seed. Then comes your date palm, Phoenix dactylifera. So it was excavated at King Herod's palace near the Dead Sea. And what is the record of the dormancy? It is 2000 years. Okay, so see that if the question can be twisted and asked, name the oldest seed. Okay, what is the uh, dormancy period of lupine? The record one you can write. Then the scientific name of date palm, scientific name of lupine, all different questions can be asked. Okay, please go through it. Now let's understand with the fruits. Now you understood about the ovule to seed. So seed structure is clear. Now we are understanding the fruit that is developed from the ovary. So maturation of ovule to seed is clear. How the water is reduced, how the parts are present on it. Okay, and you have understood the two types of seeds. Now we are coming to the fruits. Fruits are formed or developed from the ovary. Okay, so seed, you have already understood, it is the product, the final product of sexual reproduction. Whereas fruit, you cannot give that answer as final product over here. Why? Because you have heard about the term parthenogenesis. So it is your seedless. Here, seedless fruits are called as parthenocarpic fruits. So we'll just understand the three types of fruits. There is your false fruit. False, when I say, it is not from ovary. It is not from ovary. Okay. So here it develops from thalamus. It develops from thalamus. Other than ovary, that is thalamus, or it may be receptacle like your inflorescence. Let me show you the example here. Observe here, this is the flower actually. Before it was a flower, now it is a fruit. The flowers, you can see the seeds on it. Okay, this is a receptacle. Many flowers will be here. So this becomes a receptacle. The receptacle forms as your, here they have given the term thalamus. Thalamus on an inflorescence, it is termed as receptacle. If it is single flower, we tell it as thalamus. Thalamus is the region where the ovary is placed. So you can observe here in the apple also, ovary is over here, but we don't eat that, we throw that. We eat the thalamus. This is thalamus. Is it clear? Similarly here, the thalamus, this part we don't eat. This is the TS section, this is the LS section. So you have even seen your cashew nut. You don't eat the fruit. You are eating the thalamus here. This is the one which we eat. Of course, the fruit we eat, there is a process. Okay, you have your unique process. You have to keep it in the fire so the outer layer is broken. Then we take the nut, which is very nutritious. Okay, so that is about your false fruit. It develops from the thalamus. So apple, cashew, strawberry is your example. Coming to true fruits, definitely it is produced from the ovary and it is after fertilization itself. Okay, so it is developed from the ovary itself. So any example you can give: tomato, mango, coconut. Then coming to your parthenocarpic fruits, it develops without fertilization. Okay, develops from the ovary without fertilization. Your banana is your best example. You don't find the seeds in it at all. So this is one more view. You can see the flower. So there are many small flowers. This becomes your receptacle. You can find the word better here, receptacle. Here hypanthium, that is nothing but your thalamus, another term. Okay, so you can see when it develops into the fruit how it becomes. Just observe in your own time. So coming to your parthenocarpic fruits, which I said seedless fruits. Don't get confused with parthenogenic and parthenocarpic. So when I say carpic, we are talking about the wall layers. You remember your pericarp. Pericarp is your fruit wall. So fruit wall consists of your epicarp, mesocarp and endocarp. So you just observe here. You don't find seeds here, but your fruit wall, the pericarp, is edible. Here also you observe. Here the whole fruit does not have the seeds. Here also in pineapple, this is the banana is the best example. Okay. If the question is asked for 1 mark, what is parthenocarpy? You have to write the development of fruit from an unfertilized ovary is called parthenocarpy. How are they formed? They are induced through application of growth hormones. This is possible in your certain cases. You might have heard about seedless grapes, seedless watermelons. So those are all your induced through your growth hormones. So it does not require fertilization. It is directly developing into the fruit. So that's why they are called as seedless fruits. Your banana is natural parthenocarpic fruit. Okay, it was not induced by anything. It is a natural parthenocarpic fruit. Now we are coming to the topic, a very important topic called apomixis. So you have understood about parthenogenesis. Now there is something called apomixis. So till now you have understood fruit that is developed without fertilization. Can a seed form? Yes, it can. And that is your apomixis process. So production of seeds, remember, seeds, production of seeds without fertilization is called apomixis. I repeat, it is the production of seeds without fertilization is called apomixis. So this is a mechanism, a special mechanism evolved in Asteraceae, you know, your sunflower, your grasses, your sunflower families, all which flowers look like sunflowers, they all come under Asteraceae. Okay. Then it is actually the A type of asexual reproduction. Why? Parthenogenesis occur. So when I say parthenogenesis, there is your no fertilization, but an individual can form from that particular part. So here it is the production of seed without fertilization, but that seed can grow into a new individual. Okay. Coming to the next point, it mimics, it is copying the act of sexual reproduction. Why it is sexual reproduction? It copies. See, it produces the seeds. Production of seed means your final product of sexual reproduction. That means this apomixis is producing seeds, but actually it is not by fertilization. It is direct. Okay, it is directly producing the seeds. So this is why it says it copies sexual reproduction action. So it mimics, but it is actually the type of asexual reproduction. In some species, the diploid egg cell is formed without any reduction. Okay, reductional division, and the embryo is formed without fertilization. So I hope it is clear. The egg cell is diploid. Otherwise, in embryo sac, it is all haploid. So there are certain cases where the egg cell, the embryo sac formation remains diploid. Okay, because the nucellus, which one nucellus has to form into your megaspore, the megaspore remains still the diploid. This megaspore, without any further reductional division, it directly forms the embryo sac. To make you clear, you can see here 2n. See, you can see they have put a cross mark. They've become as aposporus, and megaspore mother cell is again 2n. Instead of meiosis, it is mitosis here. Okay, so mitosis occurs. Don't worry about the term. This is for your further studies, that is your CET point of view. So it is here you can find apospory. That's why it is gametophytic apomixis they are saying. This is your diplospory. Don't worry. So this is from your megaspore, that is still 2n. This is mother spore, megaspore mother cell, that is 2n. Okay, so we are saying it is directly entering into the embryo sac formation. That 2n state is only helping for the development of embryo. So you can see the development of embryo, heart shaped embryo. Okay, don't worry. This is not there for your exam. This is not there for the exam. The diagram is not there. Whereas the details, it is very, very important. Very important. Okay. So this is again, I'm just showing you all. Even if it undergoes meiosis, then there is your N and N formation. Okay. So two oocytes will fuse to form 2N. So these are all the genetic mechanisms that occur. These are little extra. It is deviating from your sexual reproduction. Okay. And there is, if in case it is single and does not find another oocyte, then the chromosome can replicate to form 2N. So this is how it happens to form into a diploid zygote. So I hope it is clear. So otherwise it is through mitosis only the female egg cell will be diploid. So direct mitosis. So you're getting some idea here with this particular flow chart. Okay. This is your in general, sexual reproduction in general. This is asexual reproduction in general. So I hope it is clear. This is a very important part, 5-mark question. What are the benefits availed to a farmer with the process of apomixis. So benefits to a farmer we are talking about. So imagine you are a farmer, you know about apomixis. Okay, so just let us understand the points and you will get the idea how the story goes. So advantages or your benefits. So it is helpful in cultivation of hybrids of a particular crop. So you have, imagine you have all maize. The maize when you want to grow, you are able to easily cultivate them. Okay, then it avoids the production of new hybrids. New hybrids are not formed. Okay, this helps us to keep the same quality. Sometimes the quality changes because of the genetic recombination or the cross pollination. The hybrid or the quality changes. We want to keep it as the same quality. It is a good quality, best quality. I want to keep it the same way. Even I want the seeds of the same quality. So that's how it avoids the production of new hybrids and keeps the same hybrid as it is. So the cultivation of hybrids is easy, as well as production of new hybrids is avoided. So once the seeds are ready, they need not go and buy from the farm again, from the agriculture farm. So it avoids the expenses spent on the seeds again, on buying the seeds. So this is a benefit for a farmer. Since there is no segregation of character, you are not getting any hybrid. So they have given there is no segregation of characters and the research to understand genetics. And hence there is a research. You can add here, and hence there is a research to understand genetics of apomixis. Okay, at present there is a lot of studies going on. For most of the crops they want to bring this technique, this genetic mechanism, so that it is easy to maintain that particular hybrid. Usually it happens to your different hybrids are formed. To avoid that, we are inculcating that apomictic genes into that crop. So I hope it is clear. These points are enough for your answer of benefits to a farmer. Okay, coming to your last, that is your polyembryony, occurrence of more than one embryo in a seed. So more than one embryo in a seed, that's why polyembryony. Okay, so what is actually happening here is some of the nuclear cells surrounding the embryo sac start dividing and protrude into the embryo sac, developing into new embryos. Just observe this. This is your nucellus. You can find some of the nuclear cells turning into your embryo. This is the normal embryo they are showing. This is the nuclear embryo they are seeing. This is the endosperm. So you can see the nuclear region having certain embryo. So they also either they can produce here itself or enter into the endosperm, enter into the embryo sac and develop into a new embryo. So you can understand here, either the zygote divides to produce more than one embryo or the nuclear cells will divide and enter the embryo sac. So this is actually your embryo sac that has now developed into your endosperm. This is the nuclear region itself. So this is how your polyembryony is formed. So we are done with the chapter also. I'll be conducting the exam for you all. I will let you know the date when. Okay. With the conditions I will let you all know. On Zoom app I'll be conducting the exam. Okay. So this chapter exam will be conducted. I'll let you know the date. So please be ready. So whatever questions are covered, please go through. And any doubts, let me know. Okay. Yes, thank you. Please answer these questions and submit before 10 o'clock.